Hi everyone, welcome to the Taxonomy and Systematics video. By the end of this video, you should be able to compare different approaches to classification, describe the basic principles of evolution, discuss different sources of evidence for evolution, and identify adaptations and explain their function in evolution. And this is something that we'll come back to over and over through the course of the semester as we move through the taxa. So again, this is a cladogram that shows evolutionary relationships. The smaller the V, the closer the relationship. And you'll be able to, we'll talk about adaptations that kind of occur at these divergences. So it's different adaptations along evolutionary history that separate our different taxa of vertebrates. So this should be a bit of a review. Sorry about that. The common characteristics of vertebrates. They have a vertebrae, or a backbone. They have a cranium or brain case, a skull. They have either cartilage, bone, or both, paired appendages or limb girdles, and pharyngeal clefts as embryos. So all of these are vertebrates and they all share those common characteristics, but you see variation, right? And so it's in that variation that we're able to classify into different taxonomic groups within the vertebrates. So how do we organize vertebrates? The binomial system is kind of how it all started. You go back to this guy, you know his name? Can you think of it? His name originally was Carl von Linné, and then he Latinized his name, Carolinus Linnaeus, when he came up with the system of how we name organisms, binomial nomenclature. So he actually made his name into binomial nomenclature. And this is before we had kingdom, phylum, class, order, family, genus, species. Linnaeus just came up with genus, species, and he used Latin because it's no longer used, it's a dead language. It's no longer used by people to converse. And so when languages are used by people to converse, they change over time. But since Latin is not used, it is static and it'll stay the same. And it's something that can be recognized worldwide. If you use a language that was only spoken in some places, but not others, that would exclude some. So Latin is used for scientific names. When you see or hear the word specific epithet, that just means the species name. So like humans are Homo sapiens, that's our scientific name. Homo is the genus, sapiens is the, you might say species name, it's the specific epithet. And so you either italicize or underline scientific names, capital for the genus, lowercase for the specific epithet. And if there's a name and date after the scientific name, that indicates who described the species and when. So just that should, a lot of that I'm sure is review, but this is kind of the basis for classification, this binomial nomenclature system. Later, we moved to including more groups. So not just genus and species, but then family, class, order, and so on. So if you were going to classify these critters here just based on how they look, so for centuries, classification was based on how things look. That's all. And so if you're going to classify these based on how they look, what would you key into? How would you start separating them? So think for a minute, look at the differences that you see and the similarities that you see. Maybe you'd look at whether or not they have horns or whether or not they have ears. Maybe you'd look at coloring, you'd look at teeth, eyes. There's some differences and some similarities, right? That's exactly how we classify vertebrates. So these are all canid skulls. So there's some similarities they have. Their teeth are very similar. And there are some things about their skulls beyond just the teeth that are similar and that are constant across all canids. But then within the canid group, you see differentiation, right? There's differences between a coyote, red fox, wolf, and domestic dog that are laid out here. And so getting to those, understanding the similarities and the differences are really important in classification, especially when classification is based only on morphology. But in recent decades, the past really just like 30-ish years, molecular information has really pushed forward our understanding of classification and how things are related. So there's sometimes a debate, is it better to look at morphology or to look at molecules? And most of the time now, both are used. But this is an example of how if we only look at morphology, we can get an incomplete picture. So look at these four, and based just on morphology, which two do you think would be most closely related? Or which ones do you think would be most closely related? Historically, it was thought that pigs and hippos are most closely related. They both have 4 toes. They just kind of look similar, right? They have similarities in their morphology and their behavior. And then it was thought too that these two would then be more closely related to cattle than to, say, marine mammals. But it turns out when looking at molecular data, so looking at DNA and proteins that are made with the blueprint of the DNA, that hippos are most closely related to cetaceans, to whales and dolphins. And then these two are more closely related to ruminants like cows, cattle, than they are to pigs. So of these 4, pigs are farthest removed from the others. And we only know that because of looking at DNA and that molecular data in addition to the morphology data. And so if you just look at morphology, this is how you would classify these groups of ruminants, the hippo group, the pig group, and then the cetaceans. But then when you add in the molecules, it totally rearranges things. And so now some folks recognize there's the way that we kind of name the different groups is changing now. And there's still some scientists that recognize the old way, some scientists that recognize the new way. So classification is constantly changing, which is really pretty cool. It can also be sometimes frustrating, like even through this class, each year, there's some scientific name on one of our lists that has changed. And usually it's one of you that brings that to my attention. So be on the lookout. The word systematics, I'm sure you've heard it many times before. Whether or not we have a clear understanding of what it means, I wanted to kind of go over that. So systematics is the study of the diversification of life forms and the evolutionary interrelationships of living things. So we can classify, right, based on morphology and based on molecules, but the systematics is really looking at, like, how did this happen? When did this happen? What is the change that brought this kind of divergence about? So it's getting more into the details of it. And it looks at geographic variation. So sometimes you'll have one species and it's the same species, but in different places, it might look different, be a different size. Or it could be that means there's 2 different species. So systematics looks into that, looks into distributions. It factors in principles of evolution, which we'll talk about in just a minute. Also looks at the geologic record, looking at fossils to try to understand evolution and the relationships. So we're going to talk about the principles of evolution and evidence for evolution really quickly. This should all be reviewed, but it is very pertinent to our journey through this semester as we go through the different taxa. So definitely this is review for you. Principles of evolution. Do you remember Lamarck was the guy that said, giraffes started out having short necks and they stretched their necks. So over the course of their lifetime, their necks became elongated. That is not true, right? Then Darwin and Wallace came along and presented the idea of natural selection, where it's not that over the course of a lifetime, something changes and then gets passed down. It's that there's variation in a population And the variations that allow individuals to survive and reproduce are the ones that get passed down over time. And so over time, you know, if you had short necked giraffes and long necked giraffes, and the long necked giraffes were the ones who were eating, surviving, and reproducing, over time, you're going to see more of these, because these are the ones that are living and reproducing. These are the ones that aren't. Genetic drift, bottleneck, founder effect, non-random breeding, and mutations are other things that can affect evolution. Natural selection is really the driving force that we see, especially as we move across these different taxa and across evolutionary history of vertebrates. But genetic drift is when you have a really small population, and the random mutations that happen can actually change the population, not because they're things that are helping individuals survive and reproduce, but just because the population's small. That's also what happens in bottleneck when you start out with a big population, shrink it down because of maybe some catastrophic event that happens. Founder effect is when a group goes from a big population and kind of breaks off, maybe it goes to an island or something. Again, you're starting with a small gene pool in all of these cases, and that can move evolution in weird ways that aren't natural selection. But again, oh, and if you did non-random breeding. So like with dogs, when we say, let's try this, and we do really specific breeding for certain characteristics, this happens in agriculture too. That's not natural selection. But as we look through evolutionary history, looking at the evolution of vertebrates, natural selection is the driving force between the changes. So all of the characteristics that we see in the vertebrates that we talk about, we can think about this adaptation. Why is it an adaptation? How did it help certain individuals to survive and reproduce? How did this persist over evolutionary time? And that's really a fun part of this course, I think. So evidence for evolution that comes into play as we classify organisms, homologous structures. So if you saw, knowing nothing about vertebrates, if you saw a human, a bat, and a whale, you might not have any reason to believe that they were pretty closely related as all mammals, right? But when you look at their arm bones, they have the same bones. It's just for different, they're modified for different functions. So that's really pretty neat. If you were to look at an insect wing, there's no bones in there, period, and certainly not homologous bones. And if you look at a bird wing, they're bones, but they're structured differently. So a bird wing doesn't have like the fingers coming down like that. The fingers of a bird wing are fused at the end. So homologous structures show relatedness and are a clue into evolution. We also have fossil evidence, molecular evidence, and that embryonic evidence too. So we talked about this before, that in all vertebrate embryos, you see those pharyngeal pouches or pharyngeal clefts that sometimes are called gill slits. So there's just common characteristics that we see when we look at these different things. In proteins and in DNA, we can see more things in common among individuals that are more closely related. This is pretty neat. This is kind of a map of Hox genes. Maybe you've heard of these before. There's some pretty neat research where they take flies and they mess with their Hox genes, and it makes their wings come out. They could have like feet that come out of their antenna, or wings that come out of where their antenna should be. So these are the genes that code for like body formation. That we have two arms that come out the side of our torso. Our Hox genes program that. Now, if you look at, so these are all animals, but if you look at the teleost fish and the tetrapods, they're the only ones that have multiple copies of the Hox genes. So at some time in evolutionary history, as vertebrates were branching off from other animals, those Hox genes got replicated. And what that allows for is a great diversity in body form within vertebrates. And that great diversity in body form is what we're studying through this semester. So that's a really pretty cool thing. we're going to go through fish, amphibians, reptiles, birds, and mammals. And again, I mean, you just, like these body forms, you look at them, body forms, the behaviors, vocalizations, and some adaptations for either getting prey, obtaining prey, avoiding prey, competing for mates. It's the adaptations that we're going to look at are so cool. It's so fun, I think, to look at the different adaptations as we move through the taxa, and to think about what is this for? What's the deal? But each one of these interesting characteristics you see, something about it helps the individual survive and reproduce. That's what it's all about. So make sure that you can hit these kind of 4 bullets. If you can't, go back, watch that part again. Very important. Closing. What do you call a moose that plays piano? A musician. See you in class.